Class, continue with chapter 11 here. We're going to talk about the transportation of ions across membranes, ions and small molecules. So let's talk about the permeability of these membranes and why they need active transport. And so the plasma membrane forms the barrier that separates the cytoplasm of the cell to the exterior environment. We've talked about this throughout the last couple of chapters. And then the organelle membranes, these separate the interior of the organelle from that cytoplasm or the cytosol. So in a pure phospholipid bilayer, which is a really good chemical barrier, there is some permeability and some things that are not allowed to pass through. So there are a lot of gases, such as carbon dioxide, nitrogen, and oxygen, that are small, uncharged, and water soluble. So they'll diffuse right through the membrane. There are also some molecules that are slightly permeable, and this is like water and urea, and ethanol, which is nonpolar. It is more permeable than slightly permeable, but less permeable than gases, if that's in between. So we're listing as permeable, but it is not as permeable as those gases. But it'll still be able to get through the cell. So then we have impermeable things, and these are things with charges. So we have amino acids, ATP, blah, 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 nucleic acids, all these things down here. And the reason for that is, remember, we have these hydrophobic and hydrophilic aspects to the membrane, and so things with a charge get pushed away because of the charge of those polar heads. And in addition, ions that have charges, they have a really hard time getting through as well, right? Because we have that plus hydrophilic, and then the, oops, that should be a minus hydrophobic tails inside there. So anything with a charge is going to interact with something with a like charge and not be able to get through. And then the last thing we have are these large molecules, so something like glucose and fructose. Because of their size, they're not going to be able to get through. So there's got to be some sort of aid in transportation that will allow these things on the outside to get inside. For example, we need calcium, we need potassium, glucose, fructose, although they can be made within the cell. In something like a plant, not every cell makes glucose, right? Only the photosynthesizing cells do. So the roots need to be able to take, those cells that make up the roots need to be able to take in glucose. There's got to be a way to take it into the cell, and it has to be passed from the leaves to that tissue. So there's got to be some sort of active transport that allows these things in. So for these molecules, protein or amino acids and glucose and ions to get from one side of a membrane to another, there are these transport proteins. And these form a protein-lined pathway across the membrane, and that enables things like hydrophilic substances to move through the membrane without contacting that hydrophobic interior of the membrane. So there are three main classes of these membrane protein molecules, and we're going to start here with channels. And these facilitate the movement of specific ions or water down this electrochemical gradient. And they're not gated, they're just open all the time. So these ions are very small and they're able to move across at any point. But then there are also gated varieties that open only in response to a specific chemical or electric signal. So in the case of like a potassium pump, where we want to keep a higher concentration of potassium on one side or the other, then we have a gated channel that'll stop the flow from one side to the other. If it's an ion that needs to be an equal concentration on both sides of the membrane, then we don't need a gated channel. We can just allow this natural diffusion across. So then we also have transporters, and we have three types here. We have a uniporter, a symporter, and an antiporter. So a uniporter transports a single type of molecule down its concentration gradient. So we see a single molecule going across. A symporter, that transports multiple or two or more ions across the membrane together. And the energy available from movement of one of its ions down the gradient drives movement of one more molecule in the same direction. And an antiporter is the same thing, but in the opposite direction. So an antiporter uses energy available from the movement of one ion across the membrane to drive another molecule in the opposite direction across the membrane. And so those allow, are like sodium-potassium pumps and things like that, if you have to have a low concentration on one side and a high concentration on the other side of two different ions, then they work, this pump is something that is used for that. And then we have ATP-powered pumps last. Where'd my marker go? There we go. And the energy released by hydrolysis drives movement of specific ions or molecules against the electrochemical gradient. So if it's thermodynamically unfavorable for this ion to be on the exterior, then you need energy to move it across. You can't just use natural gradient forces to move these ions. If it's pumping uphill, so to speak, or up current, because of this extra balance of ions. So let me give an example. So say we have a ton, these are calcium molecules here, and we only have a couple on this side, but we still want to pump more calcium out of the cell. Natural diffusion isn't going to cause this calcium to diffuse to a higher concentration, right? Because diffusion goes from high concentration to low concentration. And so what we'd have to have is this pump that uses ATP to push it against its will, against diffusion, out to the higher concentration. And so that's where these ATP powered pumps come in. So let's look at how some of these pumps work together in metazoans to import lysine as an end product. So first we have the sodium-potassium pump, and this is an ATP pump, so it requires energy. So as you can see down here, we have ATP that is converted to ADP and that phosphate group, and that release of energy pumps 3 sodium ions out of the cell. So let me get a different color here. So we'll circle, we'll say these 3 got pumped out of the cell, and 2 calcium ions into the cell. And so what this does is it creates a concentration gradient where sodium is greater outside and potassium is greater inside. And now we also have these potassium channels that facilitate the transport of positively charged potassium out of the cell, and it creates this electropotential across the membrane. So we had higher concentration of calcium on the inside of the cell, and now we can use these calcium channels that just have natural diffusion to allow this diffusion to occur. And when this occurs, we have this electropotential where we have negative charge on the inside and positive charge on the outside. And because of that, the third transporter, this sodium lysine symporter, remember 2 components going across the membrane together, because of this negative charge that's created on the inside of the plasma membrane, the sodium ions are going to want to go to the negative side, right? Because they're positively charged. So they'll go through this membrane to where it's negatively charged to try to balance that charge out. And in the process, they'll take lysine with it, because this is a symporter that requires 2 of these components to move across together. And so throughout this entire series of these 3 different pumps now, we've used ATP, converted it to ADP, and then down the line, we've imported lysine. This table is kind of mechanisms of transport and a good way to summarize how this transport works and some of the characteristics. So we have simple diffusion, and simple diffusion does not require specific protein. It cannot transport against the gradient, is not coupled with hydrolysis of ATP, and is not driven by co-transport. But then if we look to, for example, to look at active transport, so I'm not going to go through every one of these, but we'll look at active transport, and it does require specific protein. It doesn't just naturally diffuse across these channels. It will go against the gradient. It requires ATP, but it doesn't use a co-transport. And so then the examples here are ions, lipids, etc., and their ATP-powered pumps. So this is a good table to know to kind of classify these kind of 4 different transport methods across the membrane. So let's look at a kind of a model here of a uniporter transport, which transports glucose across the cellular membrane. So this is a side view of the full-length human GLUT1 protein, and it's in its inward open conformation here on the left-hand side. So the cut open view of this protein shows the surface electrostatic potential, which you can see these different colors here show the different kind of charges within this molecule. And on the very outside, the green, kind of slimy green color, that is the hydrophobic regions. And the hydrophilic regions are the blue on this. And so how does this work? So the outward open conformation in step 1 here, this binds to glucose, and it depends on the concentration of glucose. So if it's very low concentration, there's not as much of an affinity for it to bind. So then once it is bound, if there's a high enough concentration of glucose, then we get this ligand bound occluded conformation, which kind of is closed around the glucose and both sides are closed. And then in the 3rd step, we have this kind of release where inward open occurs, and that will allow that glucose molecule to go inside the cell. Step 4 is this kind of reset where the transporter is occluded, both sides are closed, but there's no bound ligand at this point. And then it'll reset to its open outward concentration, or conformation, excuse me, where it will wait for the next glucose molecule. So the human genome encodes about 14 of these highly homologous GLUT proteins, 1 through 14, and they all have different affinity. So they're all set up to move things based on different concentrations. So there's one that works at low concentration and then kind of ratcheted up to the 14th protein, for instance, that would work at a high concentration. So they all have different affinities, and they can all work at different concentrations. Okay, so let's briefly talk about osmotic pressure here. You've probably had this in chemistry or introbiology or somewhere along the line. I can't remember where I learned it, but it wasn't all the way up into molecular biology. But we're going to have a refresher for you. So osmosis is the spontaneous diffusion of water across a semi-permeable membrane from a solution of lower concentration, where there's a relatively high water concentration, to one of higher solute concentration, which has lower water concentration. So in this case, we have solution B that has increased solutes, which I'll write as S, and solution A has more water. And so because of this, water is going to want to diffuse across this membrane from A to B because it wants to dilute solution B to have equal amounts of water. So in addition to this, the osmotic pressure is the hydrostatic pressure required to prevent this net water flow. So if we're looking at it from a physics perspective, we need something that is going to exert force to prevent this from happening because these ions can't diffuse, so there has to be some net balance of force. We can't just lose this force, and that's what the hydrostatic pressure would be. We're not going to get into like the mathematics about it, but know this. So in animal cells, if you put them in a hypotonic medium, so a medium that is pretty much just water, water will move into the animal cells, right? Because they have higher concentration of ions and things in solution. If you put them in a hypertonic medium, such as something, a very salt water, something very high in some sort of ion or solute, water will move out of the cell. So to kind of drive home the point with this osmotic gradients, let's look at an experiment that used aquaporin in frog eggs. So normally frog eggs are impermeable to water and don't have this aquaporin, which is a membrane membrane channel for water. And so instead though, researchers injected mRNA that encodes aquaporin into the cells, so then the cells would express aquaporin in the plasma membrane, and so those frog cells or frog eggs became semi-permeable. And once they did this, they transferred those eggs from a neutral or isotonic salt solution to a hypotonic salt solution, so a solution that has very few soluble items in it. And as you can see, the bottom egg in each of these panels is the control, and it really doesn't change throughout the entire time series here. But the top egg is the one that was injected with the mRNA and now expresses aquaporin. And because they have a higher concentration of solutes inside the cell than outside, and there's this free diffusion of water based on aquaporin, you can see that after about 3.5 minutes, they swell to the point where they burst. So let's look at the structure of this aquaporin membrane transport protein here. So aquaporins increase the water permeability of these membranes. Remember, water is kind of an in-between permeability as to being able to cross the membrane, and that's largely because of aquaporin. And so they're a tetrameric protein, so they have 4 identical subunits, and each subunit forms its own channel. So as you can see from the top view here, we have 1, 2, 3, and 4 subunits, and then in the center are these pores, and those are the areas where water transports through. So in B here, we're going to look at a single subunit of aquaporin, and you can see there are 3 pairs of homologous transmembrane alpha helices, and so these are the tubes here. So you see the dark blue, pink, and then these two going the opposite direction would be a pair. And they are connected by 2 hydrophilic loops, right? So at the tops and at the bottoms, there are these chains that connect them, amino acid chains that are loops that interact with the outside of the cell. Hydrophilic, meaning that they interact well with water, and so being on the either cytosolic or extracellular side, you need to be able to interact with water, right? Because that's what's present there. And the loops bend into the cavity formed by these 6 membrane helices, and they meet in the middle to form part of this water selective gate. so as you can see here, there's kind of this little gating mechanism in the middle. So now looking at this in the context of water and the hydrophobicity, here we have several water molecules. They're really kind of hard to see in this graphic, but you can see them kind of in the background here. And they move single file across this 2 nanometer long gate that's water selective, and it separates the cytosolic media from the extracellular media, right? So it's going across this membrane. And these channels contain highly conserved hydrophilic amino acid residues. So those are what are indicated here. So we have cysteines and histidines and asparagines, etc., that are very hydrophilic, right? Because we want to be able to attract water, and water needs to be comfortable moving through this channel. And the arrangement of these hydrogen bonds and this narrow pore diameter prevents the passage of other ions. And so the way that this is designed, this kind of hourglass shaped pore, and the constituents of the amino acids that make up this pore prevent other ions and really make it specific to water. And with aquaporins, compared to like our ATP transfer and some of the other ones, there's no conformational change required. So it's just open all the time, and it's so specific for water that it doesn't really need to be, you know, an active transport or something gated, because it's very specific for water. So for you medical professionals, vasopressin is an antidiuretic treatment that regulates the kidney water reuptake. And the way that it works is it regulates the amount of aquaporin 2 that is inserted into the cell membranes of kidney cells. So that's kind of the mechanism. So if you have too much water moving one way or another across a membrane, using medications that regulate how many of these aquaporins, or aquaporin 2 in this case, that gets integrated into the kidney cell membranes, that's a way that you can kind of treat this congenital defect. So let's talk about the 4 classes of these ATP-powered transport pumps. So first we have the P-class pumps. I'm not going to read everything here. These are things for you to know, but I'm not going to spend the time to read it to you because you can read it. But generally, P-class pumps generate ion gradients across membranes. They have 2 catalytic alpha subunits, which would be here. And they are phosphorylated as part of the transport cycle. They also have 2 beta subunits that are present in some pumps, so not always present, and they're thought to possibly regulate transport. So as you can see, they are right here, and depending on if this P-class pump has a beta subunit, it might be responsible for regulation, but it's not. Not every pump has been classified to know if that is the case. So the next 2 we have are V-class pumps and F-class pumps here, and we're going to talk about these kind of together briefly. So both of these mostly transport hydrogen ions. So we can see here that we have these hydrogen ions, or protons, moving across the gradient. And there's no phosphoprotein intermediate here, as we had seen in the P-class pump. And the structures between these 2 are similar and contain similar proteins. So you can see the different subunits that are labeled here are very similar between these 2 pumps. And now, specifically for V-class pumps, the coupled ATP hydrolysis that transports these ions against the concentration gradient happens with these V-class pumps. And they generate low pH, and generally this is something that is experienced in like the vacuoles of plants, the lysosomes, and other acidic vesicles in animal cells, because you're pumping hydrogen ions into them, which increases the acidity. the F-class pumps, excuse me, work in reverse, and they use energy in a proton concentration or this voltage gradient to drive ATP synthesis, and this generally happens in the mitochondria or the thylakoid membrane. Thylakoid membrane. And so this, as you can see here, is kind of the reverse process where we are moving in the opposite direction, but we are creating ATP, while in the V-class pumps, we're breaking down ATP into ADP. And lastly, we have the ABC superfamily of proteins. So superfamily is a loosely related group, I guess, or not super closely related, but they're very similar in structure and function. In this ABC superfamily, they contain two transmembrane domains, which are labeled T here, as you can see. And they couple ATP hydrolysis to the movement of solutes. Specifically, each of these ABC proteins is specific for a specific substrate or group of substrates. So they would move ions, or a different protein would move sugars, or another one would move amino acids, or phospholipids, or cholesterol, or peptides, or polysaccharides, or proteins. So all sorts of different things can be moved across using these ABC superfamily transmembrane proteins. So let's look at some examples of intracellular and extracellular ion concentrations. So the giant axon of the squid, as you can see down at the bottom here, is widely used in studies of the mechanism of conduction of electrical impulses because it's very large, easy to obtain, you can't really use humans, etc. So if we look at the concentrations of ions within a nerve cell versus the extracellular environment, which would be the blood in this case, you can see that there are big discrepancies where calcium's a lot higher within the cell, and sodium is much lower compared to the extracellular environment, and so is chlorine ions, and calcium's higher. Oops, yeah, that's correct. Chlorine's higher, or calcium's higher on the external part of the cell, and the proteins, as you can see, I kind of covered them up down here, are this X icon here, and proteins are a lot higher within the cell, which makes sense. And then if you were to look at mammalian cells, non-neurons, you can see that calcium is much higher within the cell. Again, sodium's higher on the external part of the cell, and this is because of those sodium-potassium pumps that we need to be able to use to bring things like lysine across the plasma membrane. We see that chlorine ions are higher on the external part of the cell, and this makes sense, right? Because sodium chloride, table salt, is going to, in water, become its ions, and so you would expect that both of those would be higher on the same side of the membrane. Proteins higher on the inside, etc. So these concentrations are kept not equal across the membrane, because then we can these cells can harness those differential gradients to move things that they need inside or into the cell or out of the cell.
So let's look at a calcium ATPase operational model in the SR membrane, which is the sarcoplasmic reticulum membrane of muscle cells. And so this is important for muscle cell function. So the calcium ATPase pumps establish low resting calcium concentrations in the cytosol of most cells, so within most cells, and muscle relaxation. So after you've used those myosin ratchets, the relaxation phase depends on calcium ATPase that pump that calcium from the cytosol into that sarcoplasmic reticulum. And so they don't really show it here, but they just kind of label the outside here is where the sarcoplasmic reticulum is. So during the pump cycle, this couples that ATP hydrolysis with the transport of those ions across the membrane. As we talked about, it's an active transport. And in step 1 here, ATP, let me label step 1, ATP binds to this nucleotide binding site on the pipe's cytosolic surface. So that's right down in the inside the cell right there. And step 2 is the ATP hydrolysis of ATP to ADP and phosphate group. That phosphate is transferred to a specific aspartate residue, which is not really shown, but you can see the phosphate group there. And this forms the E1P high energy bond. And then in step 3, reduction in the free energy of this aspartyl phosphate bond powers the E1 to E2 conformational change. So this phosphorylation here causes this conformational change where you can see now instead of the channel being on the cytosolic side, it is now on the opposite side. And in step 4 then, so once that happens, the calcium releases into the SR lumen, even though the calcium concentration is much higher than in the cytosol. So this is important because, remember, natural diffusion, this wouldn't be the case, but since this is an active transport method or mechanism, it's allowed to move from lower concentration to higher concentration. In step 5, we have this dephosphorylation that occurs. So we're removing that phosphate group, and once that happens, we go back to this conformation that we started with E1, where the channel is now back on the other side and not releasing into the lumen. It's back on the cytosolic side. So let's look at the structure of this calcium ATPase. So this ATPase will pump 2 calcium ions out of the cytoplasm for every 1 ATP that is hydrolyzed, and that establishes that gradient, that active transport gradient. And so looking at the E1 state on the left, this is the high affinity state where there are 7 oxygen atoms that surround these calcium ions in the middle here, and that's between these 4 alpha helices that are these purple. We're kind of looking at the top down here, purple ribbons there. And then in the low affinity state, when things need to be released, then the conformation changes. As you can see, things get a little more spread out, and that allows for those calcium ions to be released. And so looking down here at B, this is the X-ray crystallography structure. There are 10 transmembrane alpha helices that make up this transmembrane part here, but only the purple ones. There are 4 of them on the internal structure. You can kind of see them a little bit better in C here. Those 4 are participating in binding that calcium. And so moving on to the cytosolic domains here, you can see that there are these nucleotide binding domains that are blue, which is over on the side over here. And this binds that ATP and hydrolyzes that ATP. And then the phosphorylation domain, so once we convert ATP to an ADP plus a phosphate group, that binds to this phosphorylation domain here in green. And then the actuator domain connects 2 of the membrane's helices, and it kind of relays that phosphorylation change at the phosphorylation domain to the actual transmembrane proteins. So it's kind of the switch, so to speak. And then in C here, like I said, you can kind of see the alpha helices and kind of the ribbon structure of the 2 different conformational changes here. When there is no bound phosphate here, it is the calcium is kind of locked in the middle here, and then once hydrolysis happens, you can see a phosphate that's bound, and that changes the conformation, and it will release the calcium ions out. It's kind of hard to see these things, even for me. You can see differences, but it's not something that without labels I'd be like, oh, that's releasing calcium. So don't feel if you're not quite seeing it. Just know more about the mechanism, and the pictures are there to help you if you can see them, but if you don't worry about it. So let's look a little more in detail at the operational model for this sodium potassium pump, this active pump that moves sodium and potassium across the plasma membrane. And as a key here, I want to point out, I don't think I've pointed out earlier, that our gradients where the high and low concentrations are is listed in the first panel usually. It was on previous slides too, so if you want to go back and look at that. However, let's look at how this works. So these sodium potassium pumps move 3 sodium molecules and 2 potassium molecules for every ATP that's hydrolyzed. And the way that this works is in step 1, these sodium binding sites, which are represented by the circles here, they have a high affinity for sodium at this point. And so extracellular, cytosolic calcium, because there's a high affinity, will move in and it will bind to those high affinity sites. And at this point, the potassium sites have very low affinity. So they're not going to bind anything. Even though there's a very high concentration of potassium on the inside of the cell here, it's not going to go bind to those sites because the affinity is real low, because of the conformation it's in. So next we have phosphorylation. So we have this ATP that'll move to that hydrolysis site that we talked about in our previous slide. And similarly, we'll hydrolyze ATP to ADP and phosphorylate this conserved aspartate amino acid. And by doing that, we create a conformational change. And that flips the kind of opening. So we now have an opening on the exterior of the cell. And we've also changed the binding affinity of those binding sites. And so those binding sites that were bound to the sodium, they become low affinity and effectively release the sodium, and the sodium will drift out of the channel. And at the same time, we've increased the affinity for those potassium binding sites, all due to this conformational change that phosphorylation did. And when we do that, the potassium will flow into the tube from the exterior of the cell and bind to those sites. And because this is low affinity for the sodium binding sites, we don't bind any of the exterior concentration of sodium. And so now that we have potassium bound, there is dephosphorylation. So we get rid of that phosphate group, it moves on, and we go back to that original conformation that we started with, that E1 state. And now, since this conformational change happened, we go back to our original state for those binding sites. So the calcium binding sites have high affinity, and the sodium, I'm having a hard time with calcium today. The sodium binding sites have low affinity, or high affinity, excuse me. The potassium binding sites, K, have high or low affinity. So they're going to release. And since these sodium binding sites have high affinity now, they're going to attract, right? And so we have our release of potassium because the affinity is back at its normal low state for potassium. And we're going to start attracting sodium again and go through this process again. And so we're constantly pumping out 3 sodium and pumping in 2 potassium by this ATP hydrolysis pump. And this can go against those gradients because we're changing the affinity of those binding sites in the conformations. So let's look at those V-class proton pumps. So these were pumps that move just protons or hydrogen ions across some concentration gradient to generate electrical potential. And so there are 2 discrete domains for these pumps. There is the V1 cytosolic domain, which is this orange circle at the top here. And this is where ATP is hydrolyzed by this beta subunit, and that provides the energy for pumping those hydrogen ions. And then we have this V0 or V0 transmembrane domain, which is this kind of green square here. And this is the conduit that moves those hydrogen ions from the exterior, from the cytosol to the lumen of the organelle. And so if we have only a V-class pump, which is an example A at the top here, what happens is we accumulate those protons on the inside of the lumen, and that creates this electrical potential where we have a high concentration of positively charged ions on the inside of the lumen, and then these chlorine ions that are negatively charged on the external. And so there's an electrical potential. It's not an active force at this point, but there's different charges across this membrane. But now if we were to combine that pump with a chlorine channel, this chlorine channel facilitates the transport of chlorine ions through the membrane and gets rid of the electrical potential. There's no difference in charge across the membrane anymore, but the accumulation of these ions causes a lowering in pH. And so if you've done chemistry, you know that HCl is hydrochloric acid, so you can think of this as its and when you put hydrochloric acid in water, it dissociates into the two ions, but essentially it's like adding hydrochloric acid to the inside of the cell. So you're going to lower the pH and make it much more acidic. And so they have kind of a couple different functions. They can create electrical potentials, and they can alter pH depending on if they're coupled with these chlorine channels. So now we're going to look at this ABC transporter family. In our example here, we're going to do ABCB1, which used to be MDR1, which MDR stands for multi-drug resistant transporter protein. And then once they found it was part of the superfamily, they've renamed it. And so this is the first eukaryotic ABC protein found in drug-resistant cancer cells. And so what this protein does is it uses energy from ATP hydrolysis, like our other membrane transporters, to export a variety of drugs from the cytosol to the extracellular medium. So drugs that were used for cancer are actively being passed out of the cancer cells by this protein, which becomes a problem. And so these drugs or compounds are primarily planar, so very flat lipid-soluble molecules with one or more positive charges. So they all have this similar function because of how they're designed to attack cancer cells. Well, that also makes them easy to export for this protein, which is an issue. And so these drugs bind to the same or overlapping sites on the protein and are transported out. So similar drugs bind similarly. And tumors derived from these MDR expressing cells some examples are liver cancer or hepatomas, and they're frequently resistant to virtually all chemotherapeutic agents, so which makes it difficult. So you'd have to rely on something like radiation instead of chemotherapy. So looking at these, the ABCB1 protein here in A that we see, it contains a ligand binding site, which is indicated in black here, which is an interesting color choice. And it's close to where the leaflet-leaflet interface is, so these would be the leaflets. So it's kind of in the crutch of where these two leaflets come together, and that is in the center of the cellular membrane. And the transport binding cavity is alternatively exposed to the cytosol and to the exterior, just like our other pumps, and it depends on conformational changes. And so down in B, we can kind of see this conformational change where when it's in its cytosol stage, we see that we have our cancer drug, for instance, here, and it can go and it can access that binding site kind of between those two leaflets, and it will bind here. And then, similar to our previous pumps, hydrolysis of ATP to ADP causes a conformational change and release of those drugs out of the cell into the exterior. Now these transmembrane domains or proteins are not specific. They weren't like evolved just for cancer drugs. It's just kind of a byproduct. And so a hydrophilic ligand can bind directly to the cytosol when the opening is facing the cytosol, so like in our first example on the left here. And a hydrophilic ligand can also bind to the binding site when it is facing the exterior. And so depending on the way it's facing, you can bind these different ligands, and it's not as specific as something, say, a aquaporin or something that only allows certain things to go through. These can bind a wide variety of ligands. Like I said, multiple different drugs that have similar structures, but they're different drugs can all be exported by these ABC family, superfamily proteins. So as stated before, this ABC family is a superfamily, so there's a lot of them, and they're also functionally and structurally related. But here are some examples. So we talked about MDR1, which is that drug resistance, and MDR2, which is another drug resistance protein, and they've since been renamed to ABCB1 and ABCB4. But this kind of shows where they are. So MDR1 is found in the kidney, adrenal gland, and brain, and then MDR2 is found in the liver. And some of these are can cause diseases you are familiar with. So the CFTR, which is an ABC superfamily protein, it transports chlorine ions. That is a disease, or that is a mutation that causes cystic fibrosis. Some of these other diseases I've heard of Tangier's disease, but some of these other ones I'm not as familiar with. I am not a human biologist. If there were plant diseases, I might be more familiar with them. But as you can see, these are very important proteins, and changes in them can lead to congenital mutations or defects within a person. So let's look at this cystic fibrosis example that we had. So there is a ABC protein called cystic fibrosis transmembrane regulator, and so CFTR, as you can see labeled up here. And what happens is this is a chlorine pump that moves or allows the passage of chlorine from the external side of the cell into the cytosol. And so what happens is, for the channel to open, we have this ATP that binds to both the A1 and the A2 intercellular cytosolic domain, and we have a second ATP that comes in, and it binds to the second binding site. And by doing that, it's kind of like a pair of scissors, right? So you pull one side together and it opens the other side apart. And so these two come together, and that opens this channel. And this allows that diffusion of the chlorine ions into the inside of the cell, into the cytosolic side. And so if this second, the lower of the two ATP binding sites, if the ATP there is hydrolyzed, then it becomes ADP, and that results in the removal of that nucleotide, and what we have is closing again. So because there's nothing bound on that lower side, then we've closed the channel. So if another ATP comes in, then we open it, and so it's kind of an on-off switch, right? And so in 67% of cystic fibrosis cases, there is a deletion in a residue which causes misfolding and destruction of this entire protein complex before it even gets to the membrane. So it's a misfold, and the exosome, it says this isn't right, so it just deletes it, and it never gets there. But in 5% of cystic fibrosis cases, there is a mutation in this binding site. Let me change my color because I marked it all up here. There's a change in a amino acid in this binding site, and this doesn't allow this switch to work. It gets to the membrane, but ATP won't bind there because of that point mutation, that one change in amino acids, and that means that this channel, the on-off switch, no longer works. So let's look at a potassium channel here that's not gated. And so the way that these non-gated channels work is they have this molecular selectivity filter that makes the ion channels specific for certain ions only. And so in the case of this potassium channel that we're looking at as an example here from Streptomyces lividans, a bacteria, it is a tetramer of four identical subunits make up one channel. And so a single subunit here, as you can see, is composed of two membrane-spanning alpha helices, which are indicated as the yellow here. And then this specific P helix is the selectivity filter that allows only the potassium to move through. And the central cavity, or the kind of canal that goes through, is lined by an alpha helix as well. So when you put these together, as you can see, there are these kind of alpha helices that kind of form that channel there. And compared to a gated potassium channel, which we're not showing here, a gated channel has additional domains that will act as kind of open and closing domains, similar to what we saw with the ABC family, where a phosphorylation or some sort of stimuli like that will cause the gate domains to open or to close. Okay, let's look at how this selectivity works. And so if we're going to look at the potassium channel, and we're going to look at why it will allow potassium but not sodium to pass through it. So when these potassium and sodium ions are in solution, they are hydrated, right? So water molecules come and they bind to the ions because they have polar ends, right? Negatively and positively charged ends, just like these ions do. And what happens is when the ion is trying to pass through this pore, they lose their binding to all the water molecules. So they become dehydrated. And instead, they form those similar bonds with oxygen of these carbonyl groups in the pore. So as you can see, all these little red circles, those are oxygens that look very similar to the red oxygens up here. And they're able to fulfill that need to bind to water, to an oxygen from a water. And so what occurs then is this is able to pass through the channel because it's able to fulfill those needs to bond to water. And there's a higher affinity for those carbonyl oxygens compared to the water, so it passes through. But the question then becomes, why not sodium? Well, the reason is because sodium is a smaller molecule, and so or a smaller element. And so when it tries to pass through that pore, which is larger, you can see that it's not able to form those tight connections that potassium can, or that sodium can with oxygen, or excuse me, with oxygen in water. And so what occurs then is very rarely, about 1 in 1,000, a sodium ion will make its way through, but would rather be bound to the water in the surrounding environment. So that makes these channels much more specific for potassium as opposed to for something like sodium or a bigger ion that would be too big to fit through there. So that's kind of how they do the size exclusion or the specificity for certain ions compared to just kind of an open gate that lets everything through. So let's look at these Symporters. And so here we have a sodium glucose Symporter. where sodium and glucose move in concert or together across one of these transporters from the exterior to the interior. And the advantage of these Symporters is that it can move certain, in this case glucose, or certain molecules across towards the higher concentration gradient. So it's kind of like a one of those ATP-driven pumps, but a Symporter can also move things in the direction of a higher gradient. And so let's look at how this mechanism works here. And so we start with this outward-facing conformation where the opening is to the exterior, and then we have simultaneous binding of 2 sodium ions and 1 glucose, and this causes a conformational change. So we now have it opened to neither side at this point, where we have an occluded conformation, and then it will change to an inward-facing conformation over time. And once this occurs, this allows the dissociation of the sodium ions as well as that glucose, and releasing of these reverts it back to the original conformation that was facing the exterior, and then we repeat. So because the sodium import is negative free energy, so it drives this glucose uphill because there is no gradient for sodium. So as you can see here, this sodium on the outside, there's a lot of it, and so I'm going to make a bunch of little dots indicating how much sodium is out here, and then on the inside it's very sparse, so it wants to go inside. So because of this, it's able to drag that glucose through the transporter because it wants to go that way. So it's kind of hiding or sneaking it in against its gradient, because on the inside we know that there's a whole bunch of glucose, high concentrations, as you can see over here on the left, compared to the outside. So that's how Symporters work. They kind of, it's like a tag along, or they kind of sneak somebody in because one thing's going with the gradient and it can sneak the other one against the gradient. So lastly here, we're going to talk about antiporters. So what this is the carbon dioxide chlorine HCO3 antiporter. And so gaseous carbon dioxide dissolves really poorly in aqueous solutions such as the cytosol or in blood plasma. So instead, carbon dioxide's converted to HCO3, and this increases the plasma's capacity to carry that carbon dioxide waste that's generated by the TCA cycle, and this will passively diffuse out of cells. And so about 80% of the carbon dioxide in blood is carried as HCO3. And so in our example here, we have this kind of systemic capillaries where we have high carbon dioxide and low oxygen. So these, we've used our oxygen, and now we have a lot of carbon dioxide present. And so the carbon dioxide gas will diffuse into the blood cell across the plasma membrane here, as you can see by this red arrow that is kind of duplicated. And then we have carbonic anhydrase, which will convert the CO2 and a hydroxide ion into HCO3 within the cytoplasm of the erythrocyte, or the blood cell. A proton, or a hydrogen ion, is released in the reaction, and it displaces oxygen from hemoglobin. And so as you can see here, it goes and it displaces it from this hemoglobin molecule, and when it does, that oxygen then releases out into out of the cell into the plasma. And then we have our antiporter, which we've been talking about. So our antiporter, which I didn't do a very good job of, let's see, can I move it up? There we go, look at that while recording. The AE1 protein here catalyzes this exchange of these chlorine ions, which is right here, for HCO3. So
they take in these chlorine ions to maintain the pH within this erythrocyte and allow the HCO3 to leave. And so, like I said, this maintains, because we don't want to pump all of our negative ions out and not replace them with anything, because then we're changing the pH of our erythrocyte, and eventually it's going to get very extreme, and because of all these generating all these hydrogen ions, but with no negative ions to counteract it. So this is Antiporter is something to maintain the pH balance while also allowing the conversion of CO2 to the more soluble HCO3 form, and then allowing it to diffuse. So that is our last example of transporters, membrane transportation, and I will see you in the next chapter.